So hello, everybody. Really excited to present this talk to you. So Josh and I work on the genomics team at Google DeepMind in London. We're two of the authors of the Alpha Genome paper, and in the next 20 minutes or so, we'll cover an introduction to this model. So broadly, our team is interested in developing AI for deciphering how DNA works. And as this audience knows, this is very much an open question. So even just understanding the molecular function of genomic sequences, let alone their kind of final downstream impact on an organism's health or growth, is already an extremely challenging problem. It's especially hard to figure out the function of non-protein coding regions, but we know that they're important. For example, the majority of disease-associated variants are actually non-coding. So what do I mean by molecular function in our context? So ideally, given a DNA string like the one in the middle, you could predict a few of its core molecular properties. So for example, that string encodes that green gene there with that particular structure of introns and exons. Maybe it's expressed highly in kidney tissue, but not in liver tissue. It exists in a specific loop of chromatin, which has a certain pattern of connections to other loops of chromatin. Maybe there's two TFs binding to those promoters and enhancer blocks there. That controls behavior. So, so this is sort of where sequence-to-function deep learning models like AlphaGenome come in. We train these types of models to take DNA sequence as input and predict various experimental readouts that reflect aspects of the DNA's function. In the example on the bottom right, it's gene expression, RNA-seq coverage. So in order to learn these types of input-output mappings, the model has to learn sometimes quite complex layers of regulatory biology encoded within DNA sequence, especially for something kind of difficult like gene expression. But if the model is good, it should be able to predict the effects of arbitrary perturbations to the sequence, and we can use it to understand non-coding variation genome-wide. So we're not the first people to do deep learning in genomics. There's other models in the space. If you have a look at what's currently out there, you'll see there's a bit of a trade-off in how they approach this problem of modeling the molecular function of DNA. One big one is this trade-off between the length of the input of the DNA sequence that the model can read in and the resolution of the predictions that it outputs. So generally, you want a longer input window. This gives the model more contextual information when making predictions. So in order to predict a gene's expression over here, you might need to look at a bit of DNA sequence 500,000 bases away. And ideally, you also want high resolution outputs. So by output resolution, I mean how fine-grained are the model's predictions. So if you're modeling a DNA string that's 100,000 bases long, you're ideally also producing 100,000 scalar outputs, 100,000 numbers, instead of producing more kind of aggregated coarser predictions for bins of multiple bases. And that's important because a lot of interesting biology that we care about, like TF binding sites, for example, or intron-exon boundaries, are actually quite on a fine scale. So we want to capture that. Another axis is this modality axis. So how many kind of layers of regulatory biology is the model capturing at once? A lot of the top models in the field kind of are specialized single modality models. For example, SpliceAI and for splicing and Orca for contact maps. This is fine. The problem is that often variants do affect multiple modalities simultaneously. And in order to do kind of that analysis, you're then having to download many different models and deal with different APIs potentially. So it's a bit clunkier for the user. So that brings us to AlphaGenome. We've essentially sought to unify several of the strengths of previous models in one model. So we take a fairly long sequence of DNA as input, 1 megabase, so 1 million base pairs. We predict thousands of functional genomic tracks from it, with many of the outputs being at single base pair resolution. We also model diverse modalities simultaneously, more modalities than ever before for this type of model, including kind of the classic hits like gene expression and transcription factor binding, but also some new stuff. We model splicing with first model to do splice junction, explicit splice junction prediction. We also throw in contact maps in there for both human and mouse. So yeah, a bit more detail on how AlphaGenome works. So we train the model on publicly available human and mouse data from major research consortia, such as ENCODE and GTEx, Phantom5, and 4D Nucleome. So as I mentioned, we take 1 million bases of DNA as input, and we predict around 6,000 tracks for human, around 1,000 for mouse, covering 11 output types. An output type is kind of like an assay, like RNA-seq, and modality is kind of a layer of regulation. So gene expression is a modality, and that's covered by RNA-seq, CAGE, and ProCap, for example. So we do a bit of engineering to make the training on such long sequences possible. So we don't sort of train on the whole DNA sequence at once. We do this kind of spatial partitioning. We break the sequence up into 8 segments with a bit of overlap, send those to different devices, different TPUs, which then intercommunicate in the transformer layers. So we can have a look at some of the example predictions of the model. This is now figure 2 of the paper. So each kind of pair of tracks, obs, pred, observed and predicted. So these are sort of like the observed genomic tracks and the predicted tracks in a held out region of the genome that the model hasn't seen before during training in the HepG2 cell line. And this slide tries to make a bit of a visual point, but generally those pairs of tracks look similar. So the model does a reasonably good job predicting experimental readouts for new DNA sequences. If you zoom in a bit on the right-hand side, this is a region around the LDLR gene. So we plot some splicing-related predictions, and you can see what they look like. So keep in mind the model doesn't know anything about kind of gene structure a priori, like we don't feed any of this information in, but you can see that the splice acceptor splice donor predictions align quite nicely with the annotated exons. Also, you can see what the splice site usage and the sashimi plots for splice junctions look like. And then finally, the RNA-seq head shows nice concordance with the splicing outputs as well. So onto a more quantitative view of AlphaGeno's performance. How well is the model performing on various tasks compared to existing models? So on genome track prediction on held out sequences, like the ones you saw on the previous slide, we found that the model outperforms the existing best-in-class model on 22 out of the 24 tasks across various modalities, including actually specialized single task models, which is really nice to see. And I think that's it's maybe a slightly deep point. Like I found it really nice. It's not kind of obvious that you can keep adding more output hits, more modalities to the same model, and not degrade on performance on a key metric like gene expression. I think it's really cool. Hopefully, people will kind of put more of these things into the same model in the future. Right, so we're happy that we do well in genome track prediction. That wasn't kind of our main focus. We really see it as more kind of a requirement to do what we really care about, which is variant effect prediction. So to predict the effect of a variant, what we do is we use the model twice. We predict outputs for DNA sequence with the reference allele inserted into it and the alternate allele, so ref and alt in blue and red. And then we can see how inserting that alt allele is changing the model predictions and sort of summarize those differences, alt minus ref. In this first example, an example from the paper, we see that the alt allele in red is predicted to decrease the RNA-seq profile of this APOL4 gene in colon tissue. So we're saying the model thinks this variant will reduce that gene's expression in this context. And this is true, that the sort of truth is the top row and the prediction is the bottom row. Here's an example of a variant effect splicing prediction. So this is a variant that's predicted to cause an exon skipping event. So there's kind of a middle exon there, and in blue you see that it's included. But when you introduce the alt allele in red, you see that it sort of skips over it entirely. This is also captured in splice site usage predictions and the RNA-seq predictions. They don't always line up perfectly, but in a lot of cases they do. Right, so I think this is the most important result of the paper from me. So how does AlphaGenome actually do across many diverse variant effect prediction tasks gathered from the community? So we found that the model matched or outperformed the best-in-class external models on 25 out of 26 tasks, including, again, specialized single task models, such as Pangolin, for example, and Crumbly Peanut. Not 26 out of 26, unfortunately, but we'll get them next time, hopefully. As an example evaluation, we can look at one of the rows in this table, the EQTL effect size prediction. So this task is asking for known causal EQTLs, what's the effect size of the variant? So what's the beta posterior coefficient of these SUSY fine-mapped variants from GTEx? Or to what extent does it increase or decrease a gene's expression? Can you predict that magnitude? And this is quite a challenging task, but we managed to achieve certain performance here, both for SNVs, which is the first kind of block of bars, and also for indels. But although there's not that many indel EQTLs, I think it's 8%. One thing to note is that the model does natively support indel variant scoring, or really any kind of variant that you can fit within a megabase. It's quite flexible. That said, distal variants are still extremely challenging. If you segment by distance from target gene TSS, everybody does more and more poorly, essentially. We're just about ahead, but we definitely have not solved that distal regulation problem. Right, so finally, I wanted to go into kind of an example use case. This is one of the case studies that we have in the paper. So here we examine mutations in kind of 3 groups of yellow affecting the TAL1 gene, which is involved in a disease called T-cell acute lymphoblastic leukemia. This is a rare form of cancer. These are rare mutations. What we did is we scored all of these variants with the model, the SNVs and the small indels, and we wanted to see what would the model say about that. The known mechanism here of the disease is that all of these variants increase TAL1 expression in certain cell types. So, so here you can see some plots. The variants are in gold, as they were before, and the length-matched background variants are in gray. And it's really cool to see that the model is really sort of saying these true oncogenic variants have much stronger predicted effects than all of these matched background variants. Yeah, so the other nice thing is that the correct cell type is highlighted by the model. Also not always the case, but is the case in this example. So because the way our variant scoring works is we score the effect across all cell types automatically. So if you then rank the cell types essentially by their score or their p-value, it's nice to see that across the 4 different categories of cell types and tissues we have in the model, the sort of correct cell type for the disease pops out. We spent a lot of time on ablations in this paper to try and understand which parts of the model and the data are really responsible for driving the performance versus which parts you can kind of cut away and not make much of an impact. So for example, like how important is longer sequence length really? How important is it to have all these different modalities in one model? I won't dissect these figures, but essentially the answer depends on which specific task you're interested in. So for example, if all you want to do is accessibility variant affect performance, you only actually need to train an accessibility model. You don't need other modalities. Whereas if you're doing something like expression variant affect prediction, then the model does benefit from training on many modalities. Finally, I just wanted to highlight some limitations in the model. So we have made progress on this tissue specificity problem, which is how well the model can predict differences in signals across cells and tissues. But this is really hard and more research is required. Same for this long-range interaction problem. We have this issue of false negatives in variant effect prediction. So although kind of benchmark-wise we're state-of-the-art, it's still a bit troubling that the model does predict 0 effect for a lot of variants where we're reasonably sure there should be some effect. Species coverage is still limited, human and mouse only. We didn't personally benchmark on personal genome prediction. I know other people are starting to do this in the field. And then finally, I mean, this audience knows this, but this model is really kind of a sequence to molecular phenotype model, not like a magic sequence to organismal phenotype or disease model. So it very much has a molecular scope. So a summary of this talk before I hand over to Josh. So this is the most comprehensive and accurate DNA sequence model for human regulatory variant effect prediction. We're taking in long DNA sequence context with high resolution outputs. Variant scoring is fast, under a second. We do some engineering tricks to make that happen. And now that we've kind of open sourced as part of publication, we think this is a really great platform to extend with new data sets, new assays, and new species. Application side, we're more on the modeling side, but we kind of are aware of some applications. I think the main one is really anything involving mechanistic hypotheses for variant functions. So dissecting GWAS loci, for example, from the open targets talk we had earlier, understanding or perhaps even helping to inform the diagnosis of rare diseases, classifying RUSs. And other use cases, so we have this sequence to function model. Can we flip that around and have a function to sequence model? Maybe. And then also genome annotation in new species. Check out the paper. We also have an API where Google funds the GPUs. And check out the tutorials and join the community forum if you have any questions. Sorry, finally, the hot new thing in AI is agents and agentic workflows. So we're kind of on this bandwagon. There's now an alpha genome agent skill as part of anti-gravity, where you don't have to learn the API anymore. You can just kind of talk to the agent in natural language and say, analyze this variant for me in this disease. And it kind of makes plots for you and generates reports for you, does the ISM to identify motifs. And yeah, on to Josh.
Thanks, Natasha. And Just in the last few minutes, I thought I'd talk to this audience specifically a little bit about what's next coming from us. And the announcement is that GDM is getting interested in data generation. As you saw in Greg's talk earlier, new data sheds light on both the strengths and sometimes the weaknesses of models. And that really helps us prioritize which components that we should be paying attention to. And as a first step towards our interest in data generation, we've partnered with the Sanger Institute to announce a new AI Genomics Data Generation Consortium anchored at the Sanger. This was sort of launched and designed in parallel to FGX, but what I've been learning through this amazing symposium is the parallels and the overlapping interests in terms of data generation, experimental design, and how to make this all accessible. So we'll be hanging around after the closing remarks, so please come find us if you're interested in talking more and learning about this opportunity. And so finally, yeah, thanks to the wider team, especially our team lead, Gia Ausik. And finally, I think Natasha and I will take some questions. But before that, if you haven't heard already, there's an amazing symposium led by Gareth at University of Exeter coming up in September. So if you haven't heard about it, please check out the QR code and you can learn more about how to use AlphaGenome and other tools in a practical setting. Okay, we'll take up questions.